So I was asked to do a video on this question because a student was struggling with it. So let's have a look at the question. So what we can see quite clearly is they've given us some information here. They've talked about this FURA 2, which is a fluorescent dye, and that fluorescent dye is going to be used to measure the concentration of calcium ions inside the cell. So this is important, the fact that they have actually identified that these calcium ions are going to be inside the cell. So for us to understand that this molecule needs to get inside the cell, it therefore needs to get across, which they're asking us about, the cell membrane. Now the question says using the information in the figure, and this is important whenever they say that, use the information in the figure to explain why something may not be able, in this case, this molecule is unable to cross the cell membranes. So that means they've given you some hints here. So let's just look at the picture and identify what they've told us. Well, they've shown that we have lots of covalently bonded atoms being held together. They've also showed us that we've actually got a lot of areas that are charged. So because we know that this molecule is a polar molecule, so that's the first thing we can note. It is a polar molecule. Because we know it's a polar molecule, that therefore tells us that we are going to have to consider the fact that our membrane actually is made up of a phospholipid bilayer. And that phospholipid bilayer has got the hydrophilic heads on the outside and the hydrophobic tails on the inside. So we know that we've got our hydrophilic heads, we've got our hydrophobic tails, we know that we get a phospholipid bilayer, and because of that, that therefore means that they arrange with the heads on the outside and the tails on the inside. Well, this molecule is a polar molecule, which therefore means that it is hydrophilic. And because it's hydrophilic, it will not be able to cross the phospholipid bilayer because of that hydrophobic region. So that's a way in which they could expect you to answer this question. The other thing is there's lots of atoms being held together. It's quite a large molecule. And so therefore, the only way it's going to get across the phospholipid bilayer is through using a protein. Now this could be a carrier protein. So that's one way in which we could answer this question. Okay, so to carry on this question then, so they've already told us about the FURA 2 fluorescence, and now what they're telling us in the question is that the FURA 2 was injected into a single sensory neuron that was connected by a synapse to a relay neuron. So they're going to assess my knowledge of what happens when an impulse gets to the end of one neuron and how that message is then passed on to the next neuron. They've also told us that the FURA 2 fluorescence inside the neuron was observed using a confocal microscope. Now, just to note, this has been removed from the specification. So do note that you will not be asked a question like this before. However, in the previous specification, you did need to know about the confocal microscope. So I'm going to ignore that question because it won't be asked again, but I am going to move on to the next bit of information they tell us. So they tell us that the sensory neuron was stimulated electrically and the FURA 2 fluorescence in the synaptic bulb was measured. And at the same time, an electrode is going to record the membrane potential in the postsynaptic neuron. So let's just remind ourselves about a synapse. We should know that we have, when we look at our neural communication, we should know that there is always going to be a neuron, a presynaptic neuron. Now, they told us in the question that this was a sensory neuron. And they also told us that there's going to be a postsynaptic neuron. So in the question, they told us that this was a relay neuron. So we have our presynaptic neuron, which is a sensory neuron. We have our postsynaptic neuron, which is our relay neuron. And we also have a synaptic cleft, which goes between. So what we're looking at is what happens when an electrical impulse is sent down through the sensory neuron, and then what happens to cause the postsynaptic neuron to then pass that information on. So they're assessing that you understand that your presynaptic membrane is going to be depolarized. So what we've got is this depolarization of our presynaptic membrane. And when we get to this part here, what we actually find is that we have some voltage-gated calcium channels. And those voltage-gated calcium channels are going to be activated to open, and then calcium is going to move into our synaptic bulb. Now, when the calcium diffuses in, what that will do is those calcium ions will cause the vesicles containing the neurotransmitter to move towards the membrane. And when they move towards the membrane, that will result in exocytosis occurring, where our neurotransmitter, acetylcholine, is going to be released into the synaptic cleft. Now, that acetylcholine will then diffuse from a high concentration to a low concentration across the synaptic cleft, and it will bind to receptors on the postsynaptic membrane. So these are the receptors, and those receptors are actually found on sodium channels. And if there is enough of a stimulus from the acetylcholine being released, that will result in the receptors binding with the acetylcholine, the channel opening, and sodium will then be able to move into the membrane. And if there is enough depolarization of the membrane and it reaches the threshold value, what will happen is that will trigger an action potential. If the stimulus is too low, if there is not enough of a stimulus from the acetylcholine, that will then result in the depolarization not reaching the threshold value. No action potential occurs. A synapse is a way in which we can filter low-level stimuli. So let's look at what the question actually asks us. But let's remind ourselves about the secretion of acetylcholine from the presynaptic membrane. So one of the things for us to remember is, as we stated before, our calcium ions are going to diffuse into the synaptic bulb. Those calcium ions are going to cause those vesicles to move towards the membrane. And because the vesicle is made of a biological membrane, which is the same structure as the plasma membrane, these can then fuse together, and therefore the vesicle becomes part of the plasma membrane when it releases the neurotransmitters out into the synaptic cleft. So it's important to note that we need to think back to module 2.1, where the cytoskeleton requires ATP to move the vesicles along the cell through that cytoskeleton towards the membrane. So do note the interlinking topic areas. Now let's go back to the question. So they told us some information in the question, and what they also did is they gave us a set of results. So here we have where we can see our strength of our electrical stimulus. We've got our FURA fluorescence in the synaptic bulb, and we've got our postsynaptic membrane potential. So these are really important parts that they've given us in a table of results. We can see there's low, low, minus 60, medium, medium, plus 40, high, high, plus 40. Now, they've given us some more information where they've told us that the intensity of the fluorescence is proportional to the concentration of calcium ions. And what they've also said is that the scientist concludes that the change in the concentration of calcium ions in the presynaptic neuron is what caused an action potential in the postsynaptic membrane. And they've asked us to evaluate and explain the scientist's conclusion. So the minute I see evaluate, I automatically remind myself that I want to give information that supports and information that does not support. So I always want to give arguments for and against any time I see an evaluate question. So let's just look, and it has said explain, which means give the science of what is happening. So let's look at that table in a bit more detail then. So they've told us that the strength of the stimulus in that electrical stimulation is low. If it is low, then the fluorescence, there is a low fluorescence in the synaptic bulb. And what they've also mentioned is the highest membrane potential in the postsynaptic neuron is minus 60 millivolts. So because I know that that's minus 60 millivolts, that tells me that that depolarization of that postsynaptic membrane has not reached the threshold value. Therefore, there is no action potential in the postsynaptic membrane. So that's the first thing that we note from the table of results. Now let's look at the next one. When I have a medium electrical stimulation, I therefore have a medium fluorescence in the synaptic bulb. I've got a plus 40 millivolts, so I have reached the threshold value. I have depolarization occurring, and it's about to go through repolarization. So therefore, an action potential has been triggered in the postsynaptic membrane. And if we look at a high electrical stimulation, we have a high concentration of the fluorescence. Therefore, we can see that there is a plus 40 millivolts. We've reached the threshold value and action potential has been triggered. So therefore, let's look back at the conclusion that the scientists have given us. So the conclusion that scientists have given us is that the changes in the concentration of calcium ion in the presynaptic neuron cause an action potential in the postsynaptic membrane. Well, if we look at this then, what we can clearly see is when we have a low stimulation, we also have a low concentration of FURA. If we have a medium, we have a medium of FURA. And if we have a high, we have a high of FURA as well. So therefore, we can quite clearly see that the strength of the stimulus does have an effect on the concentration of the calcium inside that synaptic bulb. So that is a way in which we can conclude the validity in that conclusion that the scientists have come up with. Now, the calcium ions are changing from low to medium or high, and there is a difference in the postsynaptic membrane. So we can quite clearly see that when we have a low stimulus, we do not have an action potential being reached. However, when we have a medium and high stimulus, we do have an action potential triggered. So if we're going to explain the scientist's conclusion, we have to think about the fact that when there is a medium and high stimulation, the calcium ions are going to move into the synaptic bulb. Well, they also moved in when there was a low stimulation because there was a concentration of calcium, because there was a fluorescence of calcium. So an action potential in the presynaptic neuron causes the calcium channels to open. Calcium is going to move in, and that calcium moving in is going to cause the release of the neurotransmitter. That neurotransmitter is then going to bind to the postsynaptic membrane, and it is going to depolarize the membrane of the postsynaptic neuron. And it depolarizes each of them, but it depolarizes them at different levels. So when there is a low stimulation, there is a depolarization that does not reach the threshold value. When it's a medium and high, it does reach the threshold value and an action potential is triggered. So remember, evaluate is about supporting and not supporting. And this is where we can say that when we look at the medium to the high, that change in the concentration of calcium ions in that presynaptic neuron causes an action potential in the postsynaptic neuron. When you change from medium to high, there is no change in the membrane potential of the postsynaptic neuron. And that is because you need to remember that it's an all or nothing response. As long as that threshold value has been reached, then that will trigger an action potential. So it doesn't matter if that stimulus is medium or that stimulus is high, there is enough neurotransmitter released to therefore depolarize the postsynaptic membrane to the point at which that threshold value was reached. So it is important to note that whenever you want to evaluate, you need to give pros and cons. I hope this has been helpful.